Hello, students. So, today we are going to start the fourth and the last colligative properties, okay, and which is the osmotic pressure. So, before moving to this um, osmotic pressure, first we will try to know that what is osmosis, okay. So, see, the definition of osmosis is that in osmosis what happens, there is movement, okay. So there is movement of what, there is movement of the solvent molecules, the pure solvent molecules, okay. You just focus on my words, whatever I am telling. So the osmosis is nothing but it is the movement of the pure solvent molecules, okay, into the what, into the solution, okay. So the pure solvent molecules, the movement of the pure solvent molecules from the solvent side to the solution side is through a semi permeable membrane, is known as osmosis, okay. So in other words, what we can say is osmosis is also the movement of the solute molecules from the solvent molecules from the dilute solution. You know, in dilute solution the quantity of solute is less than that of the solvent, right? So the movement of the solvent molecules from the dilute solution to the concentrated solution is known as, again through a semi permeable membrane, is known as osmosis. So the definition better you write down, I have written over there, okay. So now first of all you will think what is a semi permeable membrane. So there are many types of membranes, okay, you may have heard also, but this is a one kind of membrane which allows only the semi permeable membranes. They are a type of membranes which allows only the passage or the movement of the solvent molecules through them, okay, and not the huge solute molecules through them. So I think it is clear that these semi-permeable membranes, through these are those membranes through which only the solvent molecules can pass, and the small minute solute molecules can also pass, but the large solute molecules cannot pass. The large solute molecules, they cannot pass through these semi-permeable membranes. Okay, so the semi-permeable membrane act as a barrier, okay, to the solute molecules, the huge solute molecules, okay. Now examples of these semi permeable membranes, there are many. So here I have written some of them. One is this parchment membrane, okay, you get it in market. And another one which you all are familiar with is the cell membrane of plants, okay. So cell membrane of plants is also a semi permeable membrane, okay, through which the water and the nutrients can pass through and out of the cell. That's why they can survive, right? So the cell needs some water and nutrients for their survival, right? Similarly, the waste products from the cell needs to be eliminated. So through this semi permeable membrane, the cell membrane of plants, they act as a semi permeable membrane, okay, and they allow the passage of such type of solute solvent molecules through and inside and outside of the cell, okay. So these two examples are the examples of semi permeable membranes. So whenever we talk about osmosis, it is very important to mention this point that the passage takes place, but it takes place through a semi permeable membrane, okay. So one thing you have heard, what is diffusion, right? You may have heard what is diffusion. So your work will be, you will try to find out the difference between the two, the difference between the diffusion and osmosis, okay. So they are not same, okay. Many of you may think that they are the same, but they are not the same. That's why I'm telling that you try to find out what is diffusion, then you will better, you will understand that uh, what I'm telling, that osmosis and diffusion, they are the two different things, and you try to find out. Now here one experiment is shown like this. Here you see this one. So here you can see in one beaker, two beakers are there. So not two beakers, in one beaker what is there, a inverted funnel, a funnel is kept, okay, the mouth of the funnel is covered with a semi permeable membrane. So this is the mouth of the funnel, it is covered with a semi permeable membrane and it is inverted, okay, it is reversed and put inserted into the beaker, okay, and the beaker carries water, okay, the beaker has pure water which acts as a solvent here, okay, and the inside the funnel, what is there inside the funnel, sugar solution is there, means sugar dissolved in water is present. Okay, now what you will see, after sometimes what you will see, that the level of the sugar solution slowly and gradually rises up, okay, rather than it should fall, like rather than falling, right, the level of the sugar solution in the funnel, slowly it rises up. So it clearly denotes that the not the sugar solution is passing from the funnel to the beaker, rather what is happening, molecules, the solvent, pure solvent is passing or it is moving through the semi permeable membrane from the beaker to the funnel. That's why the level of the sugar solution in the funnel is gradually rising up, okay. And so this is osmosis, this is the process of osmosis, right, because sugar solution, it is a, I have written over here, sugar solution, it is a concentrated solution comparative to water, right, which is kept in the beaker. So already I have said you, in osmosis what happens, the pure solvent molecules, it passes through the semi permeable membrane towards the solution, or what you can say, the solvent molecules, it passes through the dilute solution to the concentrated. So here the sugar solution is concentrated and the water is dilute, right. So what happens, the solvent molecules, it passes through the beaker, from the beaker to the funnel, okay, and that's why the level of the sugar solution in the funnel, it rises up. So that means the semi permeable membrane is not allowing the sugar molecules to pass from the funnel to the beaker, right, and that only I have told you, that these membranes, they do not allow the huge solute molecules to pass through it, rather it allows only the solvent molecules and the tiny solute molecules to pass through it. Some of the solute molecules passes, but not completely, right. So this was all about osmosis and the semi permeable membrane, okay. So I think that this part is clear. Now we will see what is osmotic pressure, okay. Now osmotic pressure. So osmotic pressure is represented by pi also, okay. In some books you may get some other notation also, no problem. So it is denoted by pi, some, because atmospheric pressure we represent by p, so we will get confusion if we write osmotic pressure again by p, so we represent by pi generally, okay. Now what is it, it is the external pressure that has to be applied on the solution, okay, to prevent the osmosis. So the osmotic pressure is that excess pressure which is applied to the solution side, so that the process of osmosis totally stops, okay. So I think it is clear, osmotic pressure is nothing, it is a colligative property, okay, colligative property. That means it depends upon the number of solute molecules, and it is the this osmotic pressure is that external or excess pressure which is applied to the solution side, so that the process of osmosis gets stopped, okay. So here the definition I have written. Now you see here I have drawn one figure, this I have taken from your book only. So you see these two like one tube is one this substance, this container is there, so here two inlets are there, okay. So and this is the semi permeable membrane, okay. So this is separating the solvent and the solution. So in one chamber, in the right side, solvent is kept, and in the another chamber, solution is kept, okay. So I think it is clear to you what is solvent and what is solute. When I tell solvent, that means no solute is present, only the pure solvent molecules are there. And when I say solution, that means there is solute also and solvent also, okay. So now you see what happens, you know that when we keep so you forget about this pressure now as of now, okay? So when we keep such semi permeable membrane in between solution and solvent, already I've said you the process of osmosis will start after some time, okay? So what will happen generally, what should happen, the solvent molecules should pass from the definition, as of the definition we have learned, we have learned that the solvent molecule should pass from the semi permeable, because it will, the semi permeable membrane will allow the solvent molecules only, not the solute molecules, okay? So these solvent molecules will pass from this side, because this is dilute than this one, okay? This is concentrated. Now this will pass from the solvent to the solution, right? So this passing of solvent molecules will take place, right? But what we will do now, we will apply, so this is the process of osmosis. Now we want to stop this process, okay? So what we will do, we will apply some kind of pressure on the solution side. So on the solution side we will apply some kind of pressure, external pressure, so that what will happen, you just think, if you apply a pressure at this, on this side, what will happen, the solvent molecules which were passing to the solution side will stop passing, because on this side you are applying pressure. So what will happen, whatever solvent molecules were passing will stop passing, right? Because you are applying the pressure on the solution side, yes or no? The common sense, if you are applying pressure here, the solvent molecules will not pass to this chamber, right? It will stop. So this is the osmotic pressure, okay? So that pressure which stops the osmosis process, okay, is known as the osmotic pressure. And always this osmotic pressure should be greater than the atmospheric pressure, then only the osmosis process will stop, okay? So always the osmotic pressure should be greater than the atmospheric pressure to stop the osmosis, okay? So I think what is osmotic pressure is clear to you now. Now you see, since I have said that osmotic pressure is a colligative property, it will depend upon the, it depends upon the number of the solute molecules, right? Number of the solute molecules. So what is seen experimentally, so I'll not go into details of these, all these, because they never ask you in the exam, okay? So it is, you just see it once, I have written over here clearly. So what happens experimentally, what is seen is the osmotic pressure, it is directly proportional to the molar concentration of the solute, or the molarity. C is nothing but it is the, in simple words you can say the molarity, the molar concentration of the solute molecule. So the pi is found to be directly depends upon the molar concentration of the solute, which is represented by C. Now how can you break C? C is nothing but it is the number of moles of the solute dissolved per volume of the solution, okay? So n by v, small n by v, and you know what is n? N is what, given given mass by molecular mass, and v will be given in your question. And when you like, so this is the final expression you get, okay? Pi equal to CRT. So C is the molar concentration of the solute, R is the gas. How do we get, when we remove this proportionality sign, we will get a constant which is R, okay? So this R is the gas constant, okay? Because here we are talking about the gases, sorry, not gases. So this expression, it comes from the Dalton's law, okay? From the Dalton's law of partial pressure we get this expression. So this R is the gas constant, okay? And this T is the temperature at some, so these osmosis and osmotic, all everything whatever we are talking about takes place at a particular temperature. So the temperature factor will be there in the expression, okay? Now you can break C in like this, N by V, right? RT. Now what is N? Already I've said you. So N is what, this, sorry, C is, okay, N is what, given mass by molecular mass. Here you see, this one. So okay, this one. So this expression, and then finally when you calculate it, I mean simplify it, you will get this expression, okay? So this expression will be used for doing your numericals, okay? Again I'm telling you not to go into the details of all these derivation, it is not important, right? Everything, if we go into detail, is not important for us. So only the final expression is important. The concept, so you know that since it is a colligative property, it depends upon the number of the solute molecules, and here it is the concentration, the molar concentration of the solute. Okay, so this expression will be used for your numericals. Better you note it down. Okay, so I think I have to stop here. Okay, so in the next class I'll continue, in the next lecture I will continue with the rest ones. Okay, so thank you.